morning, everyone. Good afternoon, good evening. It's been a good week since we've seen each other. I've seen some of you at the 8 p.m. in Washington as well. It's great to see you in person. So today we have the seminar for week 5. I'm going to start sharing my screen and do our usual little introduction as well. There we go. Okay. So we can probably disable the waiting room so everyone can come in naturally. So welcome back. We're halfway through the summer school. As I mentioned before, today it's the seminar session on the implementation of deep learning software from an industry versus regulatory perspective. And first we have Dr. Matthijs Kruis to talk to us. He is from Philips, the company Philips. So first of all, land acknowledgement. If you don't know which land you're on, please feel free to click on that link. Our virtual event is based in Montreal, Quebec, Canada, which is a traditional territory in Kanakakaha, and on land which has long served as a site of meeting and exchange amongst indigenous peoples, including the Haudenosaunee and Anishinaabe nations. We acknowledge and thank the diverse indigenous peoples whose presence marks this territory on which peoples of the world now gather. We encourage you to learn and discuss about the history of the land on which you reside. And then again, you might have received email from Victor Dumitru from our admin, from info@mchacks.com. Of course, you can still ask questions at mchacks2021@gmail.com as well. So please remain muted during the entire session so that our presenters are not disrupted during the presentation as well. We'll usually do a panel discussion of it at the very end. Before our first talk, we'll do the Q&A session directly after Matthijs' talk. So as I mentioned, we're halfway through the program. We finished the first 4 weeks this week. We are at week 5 on Saturday for the seminar, and currently we're over here. And tomorrow we'll have our workshop practical coding demo on fully supervised medical image segmentation with Hunter again. If you have attended previously, you'll remember him. You can bring your daughter over if you want to. And then, we have our seminar today and workshop tomorrow, and we'll have an additional workshop on deep learning framework, PyTorch, and Sebastian and I will give an example of TensorBoard and PyTorch Lightning this Wednesday. Yeah, so I'd like to introduce our first speaker, Dr. Matthijs Kruis. He completed his bachelor's and master's degree in artificial intelligence from Utrecht University and his PhD in University of Amsterdam. He's now a senior clinical scientist in CT and radiation oncology at Philips. He'll lend us some of his perspectives in the spectral CT domain and radiation oncology industry knowledge translation sort of aspects to us today. So he'll go first, and then we'll do Q&A immediately afterwards. Thank you very much. Okay.
Thanks for the introduction, Zhuxing. Actually, I also have a bachelor's in AI from. Oh, great. I did it in 2001, and back in the days, nobody thought it was going to be of relevance at any time soon. And so in 2005, I switched to a master's in medical imaging, not knowing that a decade later, more or less, AI is overtaking medical imaging, very rapidly. So that's quite funny. And also all the techniques that I used back then, or that were very immature, so I haven't in the end really worked with AI a lot. So what I'll be talking about today is going to be spectral CT, and I'm briefly, so I'm going to mainly explain what it is, and in the end, I would like to discuss how we think in Philips that this can actually help AI, but also why we need AI basically for adoption of this technique in the clinic. So I see that my Zoom. Wait, can I make this smaller? So first, I very quickly CT. CT is a technique that represents that tells you the attenuation of radiation in the body. This is usually reported back as in Hounsfield unit, but the problem with Hounsfield units is that it's not very quantitative. So if you have two Hounsfield units of different tissues, it might be that the underlying tissue characteristics are vastly different, and at the same time, due to all types of artifacts, it might also be that you have different Hounsfield units, but that actually the underlying tissue is very similar. And the way that I usually explain what spectral CT is by having a look at photography. So if you have black and white photography, you can use it very easily to recognize objects. You can recognize the shape of objects, but answering a more quantitative question becomes difficult. So for instance, if you want to know which time of the year this is, you might look and see that here is less vegetation, and you might also see that people are wearing a certain attire, and it might tell you a little bit about the temperature. But if you have color information, it becomes much more easy to recognize what's going on. You see here on the right that everything is full green, so this is in the midst of summer. Here the colors of the leaves have turned red, hence it's the fall. And here you see that the leaves are actually slightly coming back, so this is spring. And if we think about color photography, we always think that it's very old. So for instance, if I ask you, what does an image of from 1910 look like, you have this in mind, but this is actually also a real color photography, a color photo from 1910. So this is not colored in, but this is real color photography. And the way that they did that was by means of different filters. So they would make 3 different photos with different filters, and they would project them on top of each other. And that works, as you could see, pretty nicely. But this whole procedure took about 2-3 minutes per photo. And then, therefore, if you would take such a photo from a group of kids, this often was very problematic, and you see all the discolorations. And what we basically saw that the adoption of color photography became more widespread when an integrated solution came about. So the color film. So there are different solutions. I don't want to go too deep into that, but basically there are different techniques that basically acquire 2 different images. Similar to so now I'm going back to CT. So there in CT there are basically a number of different techniques that are source based, so that try to acquire different acquisitions at different moments in 2 different energies, and by doing that you can acquire a color CT, a spectral CT. But what Philips has done, what we have done is we have made a detector that actually is able to separate the signal within the detector. And therefore, the nice thing of that is that you don't need to change anything in your acquisition, but also that that the data is fully integrated, so you don't have any offsets between the 2 bit sets. The way that it works is as follows. It has two layers of two different materials. First, you get the low energy photons that are coming in, are being detected here by a photodiode. So if they come in, you get the photoelectric effect, and so basically the X-rays are being transferred into light. That's being detected, and that gives you the low energy information. The remaining information, or the remaining X-rays, they continue to the bottom layer, and they give you the high energy photons. And it's a little bit like, I don't know if any one of you have ever gone diving, but it's very similar to diving. If you dive shallow, you see all the colors, and that's because the yellow and the red lights are still there. But if you go very deep, the low energy photons, hence the yellow and the red light, are being filtered out, so everything becomes blue. So that's basically also how this detector works. And another nice thing of this is that by integrating the energy from the low and the high detector, you can recombine this and basically reduce this system back to a single layer detector. So a detector that you normally have around, and this is what we call the conventional CT, and this is basically by all means the same scan as what you would acquire with a normal scanner. So that's very nice for adoption, such that we can give that to the doctors as a sort of a fallback, and also a start where that they could use for their adoption. So normally what we get from a CT scanner at, for instance, if we scan at 120 kilo a voltage peak, is that we get a Hounsfield unit image. But if you do spectral CT, you get a whole lot of different other results. And I'll go into some of them, trying to explain a little bit what they are and how they are being used. So, what we get is a monoenergetic image. And so these are represented not in kilo voltage peaks, but in kilo electron volts, and so keV instead of keV peak. And keV peak is basically an keV peak image is basically the integration of the energy of that, but at a peak level. So you scan with a certain keV peak, but that gives you at maximum that kilo electron volt, but also it goes down all the way through the spectrum. And a monoenergetic image is an image as if you've only scanned with one energy, with one single energy, so not with a spectrum, but with one energy. We also provide a virtual non-contrast scan that takes out the iodine contrast in the image. We also provide two different results that display iodine, so the amount of contrast agent that you have injected. We can also report the Z effective, so the effective atomic number. We can also provide the electron density, as you can see here, but we can also do things like calcium suppression. So normally calcium is very dense in an image, and then if there are structures beneath that calcium, you can often not see them. By removing the calcium, you can actually start to visualize calcium or the materials that are beneath the calcium. So you have different keV images, as said. So basically, these are the what we do is we create a photoelectric effect image and a scatter image, and we know how these two are how much they contribute to the signal in water in different at different energy levels. And in this way, we can generate monoenergetic images. And as you can see here in the iodinated regions, you can see that the contrast changes when you go to different energy levels. So this is something that you could use. For instance, if you go to a low keV image, you might be able to really increase the contrast that you see in the image. While if you go to a high keV number, you can minimize the artifacts that you see. And low KV images are useful for very many applications, as you can see here. And mainly the low KV images are used for enhancing of spectral contrast. But at the same time, you can also go to a high KV image, and with that, for instance, what you can do is really reduce artifacts like metal artifacts. But as you can see here, and thereby, making especially things visible near the metal. So if you have a low KV image and a high KV image, what you can also, and this is basically what the system does, is that at every voxel you have information of these two channels, and with this you can actually characterize all the different materials that are in the body. So here you see fat, liver, and here's the aorta with iodine in it. And here's also spleen with iodine in it, and bone. So every material has its own position in this graph, and this is an image and a graph with and without iodine, and you can actually see that this line here is the iodine curve. So basically, with like every material and every everything in this graph has a certain value. So here you have fat, here you have soft tissues, here you at zero by definition, of course, you have water. You have got air all the way down here. You got bone over here, and you got iodine and soft tissue over here. And basically by looking at where if you know that soft tissue is here and then you know that high iodine is here, then you can quantify along this line, of course, what type of material you have. Same for bone. And with this information, you can generate all kinds of spectral results. For instance, you can generate an iodine map, and of course that can help you to figure out where you have perfusion, and in this case where you have a perfusion defect in the lung, something that you on conventional CT really cannot see. And for that, it's also, if you have this information, it also becomes easier to figure out that the stenosis was here. Here you can see the stenosis because it doesn't have as much iodine in this artery. So if you know that, if you have this information, also the detection of the stenosis becomes much easier. Same for finding a leakage, especially in the presence of metal. So here you see that there's actually this small leaking of a stent that was placed. So afterwards you have to figure out, is there iodine? So that is something that you can use it for. You can also use it for finding stenosis in the brain. Yeah. Oh. But there are also, so that was all for iodine, but you could also use it, for instance, for figuring out the composition of kidney stones, hereby looking at detecting of uric acid that has a specific compound, and then removing and comparing it to for instance iodine, or iodine, and this is actually not this is not iodine, this is calcium, but iodine and calcium will also look up. You will also see it on an iodine or water image. So here you know that this is a calcium stone, and this is uric acid. Another example is here in the biliary system. So if you have biliary stones, they are often very difficult to find because they have a Hounsfield density that is exactly the same as water. So here you see they both have more or less the same Hounsfield unit. But this is only the case for 70 keV, which is more or less the attenuation that you get at a 120 keV image. But actually at all the other energy levels, they are vastly different. and this is what you can see here. So you can see that they have a different set effective, the stone and the rest of the water, and that you can actually see them on the other KV images. You can also do calcium suppression. So if you have, you're interested in the material underneath the calcium, if you would make a plot of these different regions, you see that they have a different slope, and that means that the material that is underneath there is slightly different. And this you could use to visualize, for instance, a bone bruise that otherwise would be covered in calcium. And you can also visualize things like herniated discs much easier. So and this, of course, all this information can be used for by doctors to make their diagnosis. So here you have a conventional stent, conventional scan of a stent, which seem to be re-occluded, and but this is very difficult to see on the conventional image. And by having all this information, and especially the iodine in the water, you can actually see that this what the extent is of this occlusion. You could also discern between old blood and fresh iodine. Both look denser on a scan, but if you have a virtual non-contrast, you can actually remove the iodine and see. So because it was removed, it means that there was actually iodine, but you can also see that there are some old stains from old blood. Another thing that you could do is for instance, look at suspicious cancer lesions. And the question is, of course, is it vascularized, and therefore is it a tumor, or is it just a simple cyst? And if it's a cyst, it's basically something that only contains water. Now here you can already see for this case that there is actually iodine in these two lesions, hence that it's a cancer. Whilst in this case you see that there's no iodine in there, so that means that it's just a plain cyst. And this was also affirmed by a true non-contrast scan. Let's see, how much time do I have? I think I'm almost at my. So there are different types of cancers that become more where you could use it. So, for instance, one problem is often that when you get a scan, it might also be a calcification, and therefore characterization by looking at the virtual non-contrast and looking at the iodine density scan. So so we have two results. We have the iodine density scan, and we have the iodine and water scan. The iodine and water scan is a simple decomposition, and the iodine density scan is a scan where we've also removed the calcium. So if it lights up at the iodine density scan, it means that it's not calcium, and that's actually something that you can see here in VNC. It's been removed, the virtual non-contrast. So this is actually iodine, meaning that this is a tumor. There are also some cases where the contrast is actually negative, so where the cancer actually has less vascularization. So and this is an example where pancreatic cancers are known for being very difficult to see, and they have a negative contrast, and with spectral CT you can see that you can actually boost the contrast, and therefore can see the tumor much better. Yeah, you can also quantify the tumor. I don't want to go too deep into that, but one of the problems that we're seeing, or it's not a problem, but it's a feature of the system, is that you can open the scan and then you can browse through it. So you can go to different energy levels, so high, or you can go to the low energy levels, just to get the contrast that you like. And this is also, of course, very, very useful. You can also go to some of the other spectral results. So if you want to go to to iodine density, you can directly go there. You can make a delineation. You can make an analysis of a region and compare these. And this is something that, or you can go to set effective, where you can see that every material has a different set value, and therefore, yeah, you can use this really to figure out what type of material you see. You have all these spectral results, but then it's a feature, but it's also a problem, because what we often get from doctors that are not used to working with this information is like, okay, you know, what should we use? So these people really have to be trained on the usage of all this information. And so what they want to know is, what do these values mean, and what contrasts are visible, what things can we see, what things can we not see, and so getting so although this information, there's a lot of additional information that's being provided, what we see is that the radiologists really need to be trained in this. And we also provide a lot of new information, and radiologists are really afraid to miss disease, especially if it was visible on one of the spectral results, and if they haven't reviewed them, especially in the US, they are afraid to be liable for have missed certain diseases. So that's why we are very actively looking in seeing if we could solve this problem with AI. Spectral CT is, of course, an ideal candidate for AI, because for any AI system, the more quantitative and detailed information that is well aligned with each other, the more of this information you have, the better you can train it. So so that is also why we are currently working on a spectral alerts project. It's a project to use AI to aid the radiologists to digest all this information. Unfortunately, I've been trying to see if I could demonstrate something, but unfortunately I wasn't able to do that yet. So it's only this short description on that. But it's a very we're very enthusiastic about this project, and what we believe can do with this is that we can actually reduce the data load to the radiologists and provide them with insights into the data, such that they can, of course, do their work better, and make sure that they don't miss anything. I think that was my last slide.
Perfect. Thank you so much. I'll open the floor to everyone else who has a question. Does anyone have a question for Matthijs? We can. Go ahead, Strain.
So Matthijs, I'm very interested in spectral CT for investigating material composition of different tissue types. I know that there are some articles published about the subject. How accurately can you do this, you think?
It depends very much on what material you want to look at. So, sorry?
Tissue types, that we use for in treatment planning, different types of organ tissues.
Radiotherapy.
Yes.
Yes. Yeah. So that is also that's a topic that I haven't even touched upon, and that's very close to my heart, because I'm responsible also for radiotherapy specifically. So that is very. So that I think that is extremely interesting. And you can do this very so for those calculations, for instance, in protons, for proton therapy, it's very important that you get very accurate description of the materials that you go through. because it's basically proton therapy is like a game of curling, where you deposit something, a dose very precisely, but of course in order to do that you need to know where it's, or what it's going through. And if you use conventional CT, that will introduce an uncertainty in the proton stopping power of, I think, in the order of 3 or 4%, and with spectral CT you can actually lower that to below 1%. So that means that if you go through I don't know 10 centimeters of tissue, that you can reduce your uncertainty by 3 millimeters or so. So that's very very useful.
So I'm going to contact you regarding this, because
Sure.
we also work with low energy photons and brachytherapy, and as you know, low energy photon is as important.
Yeah,
so,
and the other advantage is that you can actually use for photons. So normally what you do is you take the Hounsfield unit image, you have a calibration, and then you know what electron density it is. But if there's iodine in your Hounsfield unit image, this will screw up your electron density image. Now, if you create an electron density image straight from a spectral CT, first of all, you don't need to do the calibration. But second of all, the great thing is that you the electron density of iodine is not very high. So so you can basically use a scan with iodine for your dosage calculation.
Yes, and I see some participant. Just another quick question. With the 45 keV energy that you use, what kind of doses do you deposit? Because you want to image, but you don't want to treat the patient. So what are the
So we just use a 120 kVp beam.
Okay.
And so you use 120 kVp, but then it splits the beam into 2 signals.
Okay.
And then you create a 40 keV image, but that's not. So that is that's a representation of the data as if you had scanned with only 45 or 40 keV, but you did not scan at that energy.
Oh.
Actually, we used 120 kVp, and it's automatically been segmented in the, or not segmented, split in the detector.
Okay, now it's more clear. Thank you so much. I'll email you about the rest of the questions. And thank you.
Go ahead, Hussein.
Hi, can you guys hear me?
Yeah.
Yep.
Sweet. I don't know why my camera stopped working, but anyways, so yeah, great. It's really interesting technology. So I'm just wondering if the additional information, like the Z image, it could help with making the segmentations more accurate, because in other words, like you're adding more info, like you're giving the network more information, and if you have like a multimodal, like same image but different sorts of like information given to it, like would it increase the accuracy of the auto segmentation algorithms? Would that be possible?
Yeah, I think it could, but and that's maybe I mean, you guys have basically the last time that I worked with AI models very actively, that was 2 decades ago. But like what I'm what I'm curious about is that if you yes, I think it could. On the other hand, like the data comes from 2 in the end it's still 2 channels of information. So I would also be and of course, like all the different maps that we made are representations of the data that are useful for humans. But I could also imagine that that basically any representation of the that maybe that is something that we should give to the AI models to decide. But of course, yeah, I think this information, it's a very discrete way of representation of different materials. But I think yeah, no,
thanks. Yeah, that's great. Yeah,
but I think in the end, all the information can be useful, because as I tried to show, is that in order to come to a diagnosis, otherwise the only thing that you can do is look at the shape and sort of look at the density. That's what you do in conventional CT. But the nice thing with having all this information, even though it's it's two-dimensional. You can really play with it, and you can interrogate the data and try to figure out, like, okay, if it's this, then it should take up iodine, or it should not take up iodine, or if it does, like, what could it then be? So you have much more, and of course for delineation, that's I think it's always good to have something which is very constant, and I think the Z effective for different parts of the body are fairly constant. So of course that can help with your delineation.
Very nice, thank you.
Thank you so much. And Farhad, did you have a question too, or something to add?
Yeah, absolutely. Thank you, Matthijs, for the presentation. It was really insightful. One issue, or let's say challenge that I have faced when working with multi-spectral CT actually, is that sometimes radiologists really go, and of course it's an issue of time, gets and works with 65 keV version of imaging. So is there any specific guideline or suggestion that you might have in a systematic way for, helping radiologists to go and pick, depends on different, let's say, task, pick the best energy levels?
Well, I didn't want to make this too much of a, I didn't want to go too deep into the, like, how the different systems, make their spectral or the dual energy acquisitions. But so the thing is that because we have a system that is completely integrated, what we can do is actually do the decomposition. So normally you make two images, and then two energy levels, and then you start trying to figure out what you have. So normally you do, most vendors do that in image space. The problem with doing that in image space is that you start with an acquisition, and then you have two reconstructions, and these two reconstructions are then basically conventional images. Hence, they are affected by beam hardening, and this is and then subsequently all your results will have beam hardening, and beam hardening, for instance, is much worse if you go to low KV images. Now, what we do is we take two acquisitions, but because they are separated on a detector level, they are 100% aligned, meaning that you can do your decompositions in sinogram space, in the raw data domain. And therefore, you can decompose it into two bases, so in photoelectric effect and contrast scatter, and those two values are not affected by beam hardening. So if you then make a reconstruction of a photoelectric effect and contrast scatter image, your image is very clear, and it's not affected by beam hardening, meaning that you also can go to low KV image, a 40 KV image, without any problems. And one of the main problems is, of course, that the quality of of different scanners is different, and therefore the optimal quality might be 65 for scanner A and 40 for scanner B. And if a radiologist is used to looking at a 65 KV image and trained to do that, they will stick to that, whilst like on another scanner might be actually better to go much lower and increase the contrast. So that's also an adoption problem, right? Like so the capabilities of different systems are vastly different, and also the reliability of the results that you get, and that might really that also makes adoption more difficult, because you go with what you're trained with.
Thank you.
Thank you so much for your question, Farhad. I also personally have a question, but due to the constraint of time, I think I'll email you directly, Matthijs.
That's fine. That's fine.
Yeah, great. Thank you so much for being here. Please.
Yeah, thanks for having me. And if anyone else has a question, I don't know if I can provide it in the chat. No problem.
Yeah, for sure. Well, I'll send you the Discord. Yeah, well, I'll send you the Discord channel link, and a lot of times people ask questions in there, and then people can ask you directly there if you want to. Perfect. And yeah, email address works too.
Yeah.
Yeah. Awesome.
Okay, fine.
Thank you so much. We'll see you again, hopefully next year at Astra or something.
Astra or APM.
For sure.
Okay. Bye-bye.
Cool.
Bye-bye.
All right, so we'll move on to the second presentation of today from Limbus AI. I see Joshua's in the audience, so I assume you're the one presenting. So first of all, I'll just do a short introduction about Limbus AI. So one second. There we go. All right. So the second part of the talk will be, again, implementation of deep learning software for medical imaging from an industry perspective. We just had Matthijs, who's from Philips. Now we have Limbus AI, which is a auto contouring software company that does auto segmentation on medical images. And so I'll do a quick introduction for Dr. Joshua Giambattista. Joshua is a practicing radiation oncologist in Canada who leads research and clinical data set integrity at Limbus AI. Dr. Giambattista founded Limbus AI with the purpose of improving radiotherapy treatment planning, workflow, and validated and automated AI-based tools. Dr. Giambattista manages projects with Limbus AI and clinical research partners, building, validating, and deploying AI models for clinical practice. Joshua plays an active role in managing clinical datasets quality, which requires systematic review of existing and new clinical data. He completed medical school at the University of Saskatchewan, followed by a medical residency in radiation oncology at University of British Columbia, BC Cancer Agency, and he's currently working as staff radiation oncologist at the Alan Blair Cancer Center. Oh, I see the whole crew is here. Hello.
Hey.
So I'll do an introduction of everyone. Karl is an independent researcher and entrepreneur. He's waving, holding an adjunct professorship in the Department of Physics and Astronomy at the UBC. Is that still the case? He received. Okay. He received a bachelor's and master's degree in physics and medical physics at McGill before completing a PhD at UBC in 2003 while working part-time at BC Cancer Center as a clinical physicist, focusing on potentially high impact and near-term clinical applications. He continued to explore research opportunities, including volumetric modulated arc therapy, VMAT, 4D VMAT planning, and real-time interactive planning methods for general and adaptive RT. Working with varying medical systems, the planning algorithms developed by Dr. Otto became the basis for the commercial rapid arc system, which is now used in more than 1,000 cancer centers globally. And more recently, Dr. Otto joined Limbus AI to pursue applications of deep learning and AI in radiation therapy. And then we have Carter, and he's waving. Carter leads the machine learning efforts at Limbus AI. His focus is primarily on the data pipeline and machine learning models, various image processing applications, and integration with the user-facing application. Carter studied electrical engineering at the University of Regina, followed by graduate studies in computational neuroscience at University of Waterloo. Before joining Limbus AI, Carter worked as a data scientist and machine learning engineer with a focus on natural language processing. And last but not the least, we have John. We have John Giambattista as well. He's a professional engineer with a background in electronic system engineering. Also from University of Regina. He previously worked in the industrial automation and control systems industry before co-founding Limbus AI in 2017. His role at Limbus AI is overall direction of software development, specifically of the Limbus contour application, and mostly focusing on the client side. So user interface integration of clinical workflows, pre and post processing of images prior to AI model application. He oversees regulatory affairs and works closely with regulatory and quality staff to ensure Limbus AI is complying with all applicable regulations and deploying safe and effective products to market. All right. Thank you so much, guys, for being here, and all of you guys for being here as well. So please go ahead and start sharing your screen and start your presentation. And we'll do a Q&A immediately afterwards as well.
Can you see my screen okay?
Yep.
Okay. So I was going to do the presentation today, but we have all the other founders here. We're doing a bit of a director's retreat, so timing worked out pretty nicely. So it's kind of just a product overview that I'm going through. So I'll try to kind of highlight kind of the process of working as a team to bring AI contours to market. I think we can offer a bit of a unique perspective there. So I'll try to get through the presentation with extra time for questions, because we have all the founders here, so there might be some good questions that that we could route to the most appropriate person. So we'll try to leave some extra time for questions. So thank you for doing the introduction. So here's the Limbus AI team. We're all sitting in the room here. So just kind of looking back at the process of working on AI contouring and bringing it to market, getting it through regulatory phase, and actually getting it into clinical workflows. It really took a multidisciplinary approach from the start. So we kind of broke it out into different segments here. So my main responsibility has been working on the clinical data, making sure that data going into training the models is as consistent as possible, represents the right distribution of patients, scan types, patient positions, and also represents tumors at different stages. And in terms of the AI contouring, we have Carter, who initially is bringing in his machine learning expertise and continues to do so, and works with a larger team that that we've hired and built out over the last couple of years. John's role is basically putting together the software to bring the AI contours to the clinical workflow. So developing the models is one thing, but being able to deploy them easily into clinical workflows is, an equally important aspect of working on this as a research project, or eventually bringing it into the world as an actual product. And then there's the business side of things as well, and Karl really comes in. Karl has experience with software development, clinical data, clinical side of things, and has worked on AI models as well. But he also has a very unique perspective on the business development side of things, which has been very important for us in order to bring this from kind of like a research hobby to something that we can actually get into multiple clinics, across the globe. So just kind of like an overview of what the application offers right now. So we have a comprehensive library of AI contouring models, most of which are validated in prospective research. These are built on large data sets of expert contours, and these are continuously reviewed by clinicians and our team. We've put in multi-center prospective research, which has showed high accuracy and user satisfaction, and these were important studies that kind of confirmed the approach we were taking and the clinical accuracy before we went to market. And we continue with active collaboration with multiple clinical partners. So here's kind of the overview of what the product idea is. So on the one side, when we're developing this, we have to think about like, what is Limbus contour, like what are AI contours? But I think equally important is like who is the end user, and what is the product to the user, right? Let me just so this kind of explains it. So what is Limbus contour? So Limbus contour is AI contouring models. That's the one thing. The models are important, but it's also this software package that brings those models to the clinical workflow. And there are some basic steps that we needed to take to ensure that the software was delivering what the clinics would need in order to find this useful. So it has to be fast. We thought from the start that we wanted to implement this in a way that it would be automatic, so clinics wouldn't have to take any additional steps in their workflow. We wanted it to work with any treatment planning system. We wanted it to represent existing templates clinics used. We wanted to minimize any new hardware costs for clinics, and we wanted to have the minimum product not require cloud access of patient data. So we bring all that to the clinical workflow, but the users, basically all the users are seeing are the contours. So we have to work with IT and physics in order to get this implemented in clinics. But at the end of the day, the only thing, like the end user is the clinicians and the therapists, and they're not seeing any of this in the background. The most important thing that they're seeing and need to understand is the contours themselves. So that's why, part of the principles for bringing this to market, we wanted to minimize, any additional steps that the clinicians would have to do. We want them, we want the product to be the contours for them. We don't want them to have to worry about importing, exporting scans, using new interfaces or new tools. We wanted them to just, use their existing tools, but they're starting with the AI contours versus starting from scratch, basically. And this is something that that takes some training to, especially initially when we were selling this and getting this to clinics like 3 or 4 years ago. AI contours were a new thing, but now it's more widely understood, and clinics are seeing the benefits of it. But users still have to understand the difference with using an auto segmented structure. What are the limitations of that, and how do you accurately review that in a clinical context? And so in terms of implementation, it's fairly straightforward. Basically, we intercept DICOM images after patients are scanned. We process them and we send a DICOM RT structure set to the treatment planning system. These are just some points in terms of focus of the product, in terms of maintaining easy installation, matching clinical templates, being able to run it as a background service, so clinics can basically just set it up and not have to worry about it, and having it be fast enough on local computer hardware, so it doesn't disrupt the clinical workflow. So one of the downsides to not relying on cloud access is we're limited to the compute power of each clinic. So we have to make sure the models are efficient, and we have to make sure that clinics are running the models within a time frame where it's not affecting their existing clinical workflow. So by automating it, we really are able to minimize the risks or issues there, because we're able to start contouring that scan as basically automatically as soon as the scan is sent to the treatment planning system. So by the time the users are starting to contour that patient, we've already completed and sent the structure set, as long as it's within a reasonable time frame, like I'd say under 10 minutes. In terms of our process for model creation, we kind of run an iterative process with our team. So usually we're starting with a clinical data set and it's very important that we review everything. We learned this early on, that reviewing the consistency of the labels and contours going in is one of the most important things in terms of ensuring model accuracy. And I think one of the nice things about our team is we have we maintain a very close collaboration between the engineer team, the dev team, and the clinicians. So we kind of train iterative models, and we use those models for the contour team to test out. And eventually that will speed up the amount of data we can add in or review. And then we continue retraining the models, and basically we get the models to a point where we want to validate them in clinical studies, and where the contour and review is minimized, basically to the point where it's equivalent to if we were editing a clinical contour that was coming into our data set. And then in terms of, once we roll out those models to a clinic, we want to generally have a form of clinical validation prior to that, but some clinical validation is ongoing, and we have many clinics now that we're working with closely, but also other clinics who are just using Limbus or other AI vendors to run clinical evaluation studies. It's very easy for them to do so, and we support clinics doing that. So there's lots of active research and prospective clinical validation going on now. And then we need to monitor with our own clinical users. We have to monitor how the models are actually performing in practice, because we don't have cloud access to the patient data. We really rely on qualitative feedback from clinicians to let us know how the models are performing in practice, where they're seeing any limitations, and what other models or improvements they want to see in the future. We do capture some of this data on various clinical studies, which has been helpful, but it's still very important for us to keep close contact with clinics just to get their opinion on how the models are performing, because at the end of the day, the users are the clinicians, and their evaluation of the models is somewhat subjective, and that plays into the model design, because the end of the day is to make them feel like they're saving time and to minimize their clinical burden, and basically to make them happy. That's our that's the end goal of the models. And then we also have to maintain a strong research focus to continue working on and improving existing AI models. So this is kind of what I was just talking about. So, we know all AI has inherent limitations. And it's important to educate users about this, like what are the limitations of our AI models? Main things being like patient positioning, or, very large tumors or palliative cases where, usually you can anticipate the models having potential issues. Like most of the models that we release are fairly robust to minor differences in like contrast, patient position, or things like artifact. But it's important to know, for clinicians to know that they need to always be checking the models, and there are situations where the models could underperform. Every AI model is going to underperform in certain situations. So we also have to educate our users about what the contouring practices are going into the model development, because that's basically going to determine the behavior of the models. And luckily the differences here are pretty minimal in practice, but there are some larger differences, mainly with the clinical target volumes. But it's important for the clinicians to know, like, what internal guidelines are we using to train the models, and if that differs from their expectations, then they're going to evaluate the models differently. So it's important that we educate users on that. Again, we don't have cloud access. This is run locally, so we don't retrieve any patient data. So for evaluation studies, we have to do prospective studies, and we have to connect with clinics to get their feedback on how model improvement is in the real world. And then we also have to keep an eye on model drift. So over time, as practices change, patient setups change, new CT scanners or imaging modalities, we have to ensure that the models are performing as accurately on those modalities, and that the models aren't losing accuracy or becoming biased in some way over time. So one example of this would be spacer gel for prostate treatment. There are new types of spacer gel coming out. There are some it's becoming more widespread, and maybe our initial data sets had less representation there. So that's something we had identified in a prospective study. This would have been about 2.5 to 3 years ago. And so, we use that to update the models, but those are the types of things we need to kind of look out for that could be introduced in the future. And then just looking at our product roadmap, the things we're working on are adjacent to our core strengths of basically contouring scans and fitting them into the clinical workflow. So that includes, future plans, hopefully to get into image fusion or adaptive radiation treatment. Basically, we want to leverage our strengths and integrate new AI models into other workflows that can help in the radiation oncology space. The difference is that these may be less obvious time savings, but we may be introducing new, like new opportunities or using the AI in areas that haven't had widespread use up to this point in time. So it's exciting new things that we're working on that we can hopefully have more information in the future. We do have a recent collaboration with Accuray on the adaptive side as well. So I think that was it for the presentation. So I think we have all the founders here, so definitely love to take questions in terms of most of the development and the AI side of things. I'm definitely not the expert, so good opportunity to ask the other guys here any questions.
Thank you so much, Joshua. That was wonderful. Beautiful slides as well.
Okay.
Yeah, it's amazing to see how much you guys grew in a year. Because for those of you in the audience who participated last year as well, you guys might remember Limbus' presentations last year. I especially like the education aspect of what you guys do here. It makes it very transparent, especially regarding model generalizability, educating how it's sort of built on the inside, so that user can actually learn to adjust their expectations and use it actually properly and not sort of blindly trust everything. And so I'll leave the floor open for any questions. So I see one in the chat from Rahul. He asks, so how do you get more data to improve the model in the future? I guess this can be fused to one of my own questions as well. And you mentioned that there's no hardware cost, and so there's no cloud-based thing. So what do you do with the hardware side of things? You said it's limited to the clinical ability, the hardware capacity at each clinic, but how would that actually affect the software? And on the other hand, to gather more data, to iteratively, continuously retrain your model, how do you actually retrain it from multiple different institutions? Do you use a specific framework like federated learning, or some other specific architectures that, yeah, go ahead.
Yeah, no, I mean, for the data, it's very simple. We, most of the data we're gathering is with our clinical partners, but we're not doing it federated. Like, we have to have the data centralized because it's very important for us to review the data. And I'm not convinced we can do that accurately in a federated system. Like even clinical trial data needs a lot of review when it comes down to contour accuracy. And I think we have a good process. So most models we can get to a point where they're going to perform well in multiple clinics. We have a good process for accounting for differences in setup. And it basically comes down to just including a wide variety of scans. Tumor sizes, getting in scans that are poor quality, basically anything, as long as the last thing you want is like one CT scanner with like normal patients. You want You want to have just like a wide distribution of possibilities, and then you just need to make sure that the contours on those scans are as accurate as possible. And then you have to evaluate and be prepared to update the data if you see the models underperforming in certain situations. So you have to be aware of those potential differences up front, but some of them you just kind of learn as you go. In terms like something like the prostate spacer gel, that wasn't really something we were thinking about until we actually saw that as an issue in clinical practice. And fortunately, once we identified that, it was easy to update the models to work with the spacer gel in place. But things like contrast phases, artifact, breathing artifact, those are all important. And then in terms of the compute side, John can probably explain this a bit more. I think one thing we want it to work. We want any clinic to be able to deploy this. So we've been deploying out to Ukrainian clinics over the last month, which has been John's been more involved with that. But that's kind of really highlights like what we wanted for minimum viable product. They're able to repurpose an old computer and run these models and work for them in their clinical workflow. Whereas other clinics can deploy a good GPU and get really fast contours. We still want the ability for certain clinics to run this, even if it runs in like 10 minutes, it's still going to make a massive impact on their workflow. I know you have more to say about that, John.
I think like you said, we put a lot of effort into like speed optimization of these models and kind of our whole image processing pipeline. And I think like across all our customers, like hundreds of sites on like CPU, which could be anything from like a Intel i5 to like a Xeon. I think we run a scan in an average of like 3 minutes. So for head and neck, that's like 50 plus individual structures. So pretty high performant even on older CPUs. But like you said, we have some sites that will repurpose an old treatment planning workstation from 2007, and it might run a scan in 10 minutes instead of 3, but they're still able to use the contours kind of within that like time frame of the clinical workflow where it's useful to them. And then we can always run this on a local GPU, which is becoming more common in clinics as well. So it doesn't really seem like the compute issue and needing to go to the cloud has been a big barrier for us yet.
I could add a couple more comments to that actually. So I think early on, I think Carter could comment more than I could actually, but early on we tested different neural networks, like some smaller neural networks and really beefy neural networks, and we found that we didn't really need huge neural networks that would be time consuming to get good results. So we saw the most improvement is the quality of the data. And at some point we're going to need to revisit that. But so far, I think it's still just about getting better sampling of the possible, patients that exist in the world and making sure that data is consistent before trying to put in some super fancy neural network that takes 10 times as long to compute. And another thing is in terms of the hardware requirements, because of the way our workflow is, so our software works in the background and just waits for the images to come in. So if there's a patient that is scanned, the patient will still be on the couch when the images are saved. As soon as they're saved, our software picks them up, does the contouring, and then puts the structures wherever the TPS is looking, right? So by the time the patient is outside the room and done their CT sim, the contours are ready, even if it takes like 10 minutes versus 3 minutes. So that there's no actual waiting for the contours unless the dosimetrist and treatment planner is like literally waiting at the computer while the patient's being scanned, which is rarely the case. Now that is the case for adaptive radiation therapy. So in adaptive radiation therapy applications, where the patient's on the couch and you want to potentially replan the patient, you have to get those contours extremely fast. So in that application, then you need a good GPU, and the fact that our models are not like the deep neural networks are not huge, makes it so that you can contour very quickly.
Thank you. Carter, do you have anything else to add as well?
Yeah, I think it's right to draw the attention to the trade-off between kind of like how much work you put into model architecture and how much you put into data. And we found success kind of focusing on making sure that we have good data, and then you can rely on slightly smaller neural network architecture and not have to spend as much attention on that.
I think it's also worth highlighting, like, we do like post-processing and there's other things that we add into the models. It's not just AI models for each of the structures. Like each organ is basically, has its own set of rules around it. And that's also important in terms of how the models behave. And so if there's an error in the models, we also have to kind of figure out whether it's actually the model application or a post-processing issue. So like one recent thing was, when we're training and developing the models, we're kind of working on anatomical regions to make sure the models are working in those locations. But now that we have many clinical users, there's more users doing full body scans, and we hadn't really written the post-processing rules around that. So they were encountering some issues, like femurs were running in the humerus, and some clinics were actually using it as a humerus contour. And so, just having lots of clinical users at this point, it's hard to anticipate every use case up front, but it's important to gather that feedback. So knowing that, we implemented a fix, and now the models are working really nicely on full body scans. It's just hard to foresee all these potential issues up front. So it's nice to be in clinics and have those contacts so we can make these improvements over time.
Absolutely. Dr. Anger, you raised your hand as well. Go ahead.
Yes, I have a quick question regarding, so logistically, so this is the companies, and I forgot to ask also Matthijs from Philips. So if an academic institute such as ours collaborate with you, obviously we have the patient data and so on. But how can this collaboration be handled? Because you have non-disclosure agreements regarding your code and your algorithms and models. So I'm just asking the academic center and industry collaborations currently, how are they done? Because if we provide, or we are interested to put a graduate student on the topic of segmentation, I know that there are you and many other companies who are already doing segmentation. So instead of us starting from scratch, how can we collaborate and at the same time give the student a fair chance of actually developing models and not just the company using our data? Do you understand my point here?
Yeah, do you have any thoughts on that, Karl?
Just to clarify, so you're thinking that you'd have a student, so that it'd be nice if they could have a head start, right, by kind of getting up to speed at the level that a company is capable of doing, and then potentially going beyond that in their project. And then once they do go beyond that, working with the company to incorporate that into like their commercial product. Is that
yes.
what you're describing? It's challenging. So they would have to work very closely with us, because if they develop their own models, they wouldn't necessarily be, they probably wouldn't be compatible with our system. So they would have to it would be extra work for us to make whatever they produced, compatible with our system. And also, just going back to what we were discussing in the last question, it's really the data quality that is determining how good these networks perform, right? So students working on this, it's hard for me to see where the student would be able to produce something better by just creating a different kind of neural network anyway. So yeah, I mean, I guess I don't want to sound negative, but I don't know if I'd want a student, if I was a professor, I don't think I'd get my students to work on automatic segmentation.
No, but what I mean is that so sometimes we need to do that because for our own projects, we say that in treatment, we have the treatment planning process, automation of treatment planning. We would like to do something similar, and then maybe on sites that you don't have or sites that you haven't developed anything yet. And this might be lesions, different type of lesions than cancer sometimes, for example. But and I don't mean that then the student would work on it parallel to you, but what I'm saying that traditionally when companies have worked with academia, if there is a clear transparency between the company and the academia, so that whatever developed hand in hand can be then implemented in the treatment planning system. So like a true partnership rather than just the AI companies using the academic centers for their data, but actually there is no knowledge sharing going on there.
Right. So we have, various like different agreements with institutions, and so some of them will, some institutions will just be doing research for like heart treatment. So they'll want to contour all the substructures of the heart, but we don't get into that level of detail with our heart contouring right now. So we'll work with them, and so we'll do all of the automatic contouring. We'll develop the models if they provide us the data, and then we will give them free access to those models at their institution, forever. But how it is then implemented so there could be a licensing scenario where they get a percentage of the of how much money we get from our contouring solution or something like that. We haven't done that yet, and it would have to be pretty valuable data, like almost impossible to find data for us to do something like that, because there's usually other ways of doing it. And then we already have lots of data that we could just, contour the heart substructures on, for example. It's challenging. Like I have a lot of experience working with companies from my other work, right? And so like looking at it from the other side, from the researcher side, trying to deal with the company, I think it is difficult, but there are ways of kind of like protecting yourself, like intellectual property wise, so that you can, if there's something that's patentable, you can patent it first and then talk to the company. That's what I would do. But often, this kind of thing is not really patentable with a deep neural network research. It's tough.
Yeah, I mean, it did, like when I started Limbus with John and Carter, and when we brought Karl in, we were kind of in the same boat. Like I was a radiation oncologist. I didn't know anything about coding or machine learning, and I still really don't, but I wanted to have, these AI contours that we could use in clinic. So, I was just fortunate to have a good team that could put these tools together for me. But it did take us a long time to get those initial data sharing agreements through. Like we're talking 2 to 3 years, even at our own cancer centers. But in long term, those research collaborations are paying off, and we really do need. Like I feel strongly right now that running the data centralized is important to ensure model quality over time, especially with the contour review. That's like one of the main reasons why I think our models are so good. And in order to get the clinical partnerships working, we really need to have access to the data. Like our clinical team is, and clinics that are able to do that, like we collaborate, we can get the projects done really quickly. We'll put resources on and get our contouring specialists working on the data with the clinical researchers. So it's mostly getting around those agreements, but once we get past that point, these projects end up being very successful for us and for the researchers.
Okay, thank you so much.
Okay, thank you so much, guys, for all of the answers. I think we have another question. Smai asked a question. There are other auto contouring softwares available. What are most important features that make your software different? We're all getting to the points here. So this will be our last question, and then follow that we'll have our last speaker today, Dr. Andreu Badal from the FDA, which will give us the regulatory agency's perspective on the deployment of AI software as well. Okay, yeah, please go ahead.
Yeah, I think I can take that one. So I think like our contour library, just the number of structures that we have available now, and the quality of those structures, that's kind of backed up by the prospective studies and retrospective studies that are ongoing or that we've done in the past. I mean, there hasn't really been great studies looking at contour quality across every AI auto segmentation vendor, but from feedback and kind of our, I guess our success to date, we know we're top of the line in terms of quality. So that's always a differentiator, and that, I guess it kind of plays into why we've been successful with Accurate and looking into kind of the adaptive field as well. And then beyond that, like we were talking about, just the ability and just the lightweight of the solution. Like it's deployable anywhere, pretty much any clinical workflow on any existing hardware. We don't need to go through convoluted, data sharing agreements to send our data to the cloud. We don't need to host servers in every different market in the world, so data is not leaving borders. You can really ship someone like an install link and have them running automatic contouring, within an hour or two. So I guess just quality and speed is kind of the name of our game.
Thank you so much, Sean, for your answer. And thank you so much, everyone, for all of the discussions that we had so far. So it's really great to have you guys back this year.
Thanks for inviting us.
Yeah, for sure. So now we'll move on to our last presentation of today, from Andrew. So I'll do a quick introduction of Andrew as well. 1 second. Oops. There we go. So Dr. Andreu Badal is a research physicist and regulatory consultant at the Food and Drug Administration's Center for Devices and Radiological Health. His main line of research is in the development of the GPU-accelerated Monte Carlo X-ray transport tools with the goal of increasing the use of computational modeling and simulation in the optimization of regulatory evaluation of X-ray imaging devices and medical image processing software. So today he'll share with us some of his insights in implementation of deep learning software for medical imaging from the FDA perspective as well. Please go ahead. Thank you very much. Are you? Yeah, great.
Hi. Hi, Eugene.
Hi.
Thank you very much for the invitation. It's a pleasure to be talking to you today. So can you see my presentation?
Yes.
Yes. Okay, great. So I will be talking about the regulatory perspective on deep learning and in general medical device approval process. I will center on the FDA, so that's the United States.
Andrew, you muted yourself.
You're muted. There we go.
Okay, yes. So I will give an introduction to the FDA and place that I work, and then make a overview of how medical devices are being regulated by the FDA, then explain how these adapt to the deep learning software. And finally, I would like to talk also about some of the research that I do if there is time, because you're maybe interested in this aspect. So the FDA is responsible for protecting the public health in the United States, has to ensure safety, efficacy, and security of all the drugs, biological products, medical devices, food, cosmetics, and essentially any device that emits radiation. So we are about 2,000 employees, and we review more than 20,000 pre-market submissions every year. So it's a lot of devices in the market, more than 180,000 devices, and also more than 1 million reports of medical adverse events. So that's the workload that we are handling. That's for the Center for Devices and Radiological Health, the part that focus on medical devices, and also any radiation emitting device from a CT scanner to a laser pointer. Within CDRH, I work at the Office of Science and Engineering Laboratories, which is a research part, which it's less than 10% of CDRH, it's 180 employees. And essentially what we do, in addition to research, we collaborate with the review teams to speed up and make the best review possible. And within OSL, I'm in the division of imaging, diagnostics, and software reliability. So we do research in medical imaging, digital pathology, medical extended reality, and AI ML. Essentially, AI ML has been expanding over all the research areas. So this moment, every single project has some AI ML implications. So how does the FDA review medical devices? So we use a total life cycle approach with pre and post-market activities. So the pre-market, it's based on a classification depending on risk, and the manufacturers have to show a reasonable assurance of safety and effectiveness of the devices. The devices are stratified in 3 classes. The lower risk is class 1. This is mostly exempt of evaluation and it has to follow just some general controls. Class 2 are less low-risk devices where there is some need for specific controls. And this is the applications I will talk more later about, the pre-market notifications and de novos. Finally, higher risk devices or devices that have no predicate in the market require pre-market approval, which is a more stringent process. There is also another kind of submission that we evaluate all the time, that it's pre-submissions, and that's a way for anybody to get feedback from the FDA, and it's free, and we can you can send questions and a review team will answer the questions, and that's very good to avoid having problems or new questions when the actual regulatory submission arrives, so we can discuss it ahead of time and avoid problems. Once a device has been cleared for marketing, the FDA continues some surveillance and some post-market activities, so compliance, inspections. There can be recalls if there's a problem, and also all the adverse events. There is a website where anybody can report any adverse event with a medical device, or you can search this database to know if there is any problem reported for a particular device. So the most common method for devices to getting clearance is the 510K process, the pre-market notification for class 2 devices. In this case, the manufacturer, called the sponsor, has to identify a predicate device. So it's a device that is already in the market. And then they have to show that their new device is substantially equivalent to that. And the substantial equivalence has a legal definition. It's the link in the Code of Federal Regulations. So essentially, you can claim substantial equivalence if the new device has the same intended use and the same technological characteristics, or same intended use and different technological characteristics, but these differences do not raise different questions of safety and effectiveness. And data demonstrate that this device is safe and effective as a predicate device. For devices that don't have a predicate or are class 3, the pre-market approval requires much more evidence, and it's a much longer and costly process. And when a device goes through a PMA, it cannot be used as a predicate. So any subsequent modification needs a supplement to that PMA, and any other company that has a similar device will still need a PMA. But eventually, it's possible to down-classify from class 3 to class 2 and enable the 510K process, as happened with the digital mammography that initially was a PMA, but then was down-classified. There is a third option, is this de novo classification. There is a device that doesn't have a predicate, but you can justify that it's a class 2, it's not so risky. It's possible to generate a de novo predicate, and essentially that new device becomes an existing device, and then future devices can use it as a predicate. And as part of this process, there is a special controls that are written, so there's some more stringent than a regular 510K. So to demonstrate that the device it's equivalent, you can submit really any kind of data. We review a lot of clinical data, depending on the risk, maybe a few sample images, or can be a full clinical trial. bench testing is very important too. And you can also submit like animal data, and also computer modeling and simulation. It's something we are trying to encourage, reviewing more and more computational modeling. That is, of course, very important nowadays. To help companies with all these requirements, the FDA issues guidance documents. They are non-binding, they're voluntary, but they can simplify your submission if you happen to have a guidance in your field, and this is a database where you can search them. If you make a pre-submission, we can discuss guidance for your device. If there are international consensus standards in the field, that can also make the submission more easily evaluated, and there are standards that have been recognized for FDA, and you can search, and that will simplify the process too. So with regard to deep learning software specifically, CVRH regulates it as any other software. And we define, say, two classes of software. There is software in medical devices. So that's a piece of software that it's sold with a piece of hardware, essentially. And then there is software as a medical device. So that's the official definition. Software intended to be used for one or more medical purposes that perform these purposes without being part of a hardware medical device. So sometimes there's some confusion between them. For example, if a company wants to sell a CT scanner and it has a CT reconstruction algorithm, this is a software in medical device. However, if a company wants to sell an independent CT reconstruction algorithm that can get data from different CT scans, and it's not essentially sold at the same time as the scanner, then it could be a software as medical device. Machine learning is being applied in all kinds of devices, and in my field of medical imaging, it's present everywhere in radiology. And there is a little more review compared to previous algorithms, essentially because they can have an unexpected drop in performance because they are not linear. So it's important to show some generalizability, for example. Every time a software is changed substantially, in general, a new FDA submission, new 510K of PMA supplement is required. So at this moment, in general, continuous learning would not be allowed. And if you want to retrain your code, it probably will need to submit a new 510K. Anything that is has a potential effect on the performance of the device would need clearance. So what are examples of SAMD in radiology? We're seeing a lot of image enhancement software, denoising, things like that. Automatic segmentation, they just were explaining, it's very, very, very growing field for radiation treatment, but there are also applications in 3D printing and surgery planning. Another field that is very relevant is computer-aided diagnosis, and there are 5 classes for detection of lesions, diagnosis of lesions, detections and diagnosis. There's triage software, which prioritizes, so which scans the radiologist will look at first. It's important, for example, for stroke patients. You don't want them to be waiting for the radiologist. And there's a new kind, it's acquisition and optimization. There is a new software that guides the acquisition of an ultrasound image. So that's a very new kind of device. And all of these essentially were de novos, and they have special controls. And they also have guidance documents, so you're not alone, like if you need to find out what requirements are needed. The kind of evidence that is submitted for this machine learning software has 2 components. Standalone performance is what you would typically understand for any machine learning. You have a testing data set that is completely independent of the training data set, and we want to know where the data comes from and things like that, and then show that it generalizes in different inputs and different also it needs to be tested in different locations and people with different like racial profiles, so it can generalize in the US population. And also there's usually clinical performance, which depending on the risk, maybe like just a few images, or it can be a full clinical trial. And if it's something like a computer-guided diagnosis that has special controls, then there is more constraints that are specified in the regulations. I will not go in more detail because these rules are changing very fluidly. It's a field that it's changing so fast. The CDRH created a Digital Health Center of Excellence, and I direct you there to find the latest regulations and perspective from the FDA. I would like to take out this opportunity to talk about some of the regulatory science research that we are doing. That's what the Office of Science is doing. We combine part of the time in the regulatory side and part in the research side. So FDA has a specific definition of regulatory science. It's the science of developing new tools, standards, and approaches to assess the safety, efficacy, quality, and performance of FDA-regulated products. So it's kind of different than the science that NIH, for example, would be doing. We don't do so much basic research. It's more with an intention to eventually help medical device products. And all this research we are doing, even though we go to conferences and publish papers, it has a direct impact in regulatory process because we participate in the review team. So we meet with the companies and we provide our feedback on the processes. So that's an example of a project that I'm very proud that I participated. It's called the Virtual Imaging Clinical Trials for Regulatory Evaluation, or VICTRI. And we demonstrated that it's feasible to use virtual clinical trials to reproduce an existing clinical trial. So the objective was to compare the detectability of lesions and calcifications in a mammography image or in a digital breast tomosynthesis image, which is it's a new technology that it's almost a 3D version of mammography, more or less. To do this, we wanted to do everything in the computer. So this was a study that has been done by a company in a real population, but we wanted to replicate it completely in silico. So we use a Monte Carlo X-ray transport software that I wrote, MCGPO, to create a digital twin of the real device that was used. Then we created a virtual breast phantom. We created 3,000 of them, an image in silico with this digital twin. And then we use mathematical model observers to detect the lesions that we inserted in this object. So that's an example of the virtual anatomy of the breast. We can generate thousands because it's completely mathematically defined object. And this is an example of the lesion that we inserted. This is also a mathematical model. And on the side, it's examples of the mammograms that we simulate. This is an heterogeneously dense breast. And we have 3,000 of these, and they have a very nice variability on the distribution of the fibroglandular tissue and adipose tissue. And there are lesions inserted in here. Okay. So that's the kind of images that we have. On the left is a simulated mammogram with 3 lesions marked that are difficult to see. And on the right are 2 reconstructed TBT slices. And you could tell probably it's easier to detect them here, and you can separate them in 3D. But we don't need to guess if it's easier because we can use a model observer to detect them because we know exactly where they are. And that was the primary outcome of our trial of 3,000 patients. The metric was the area under ROC curve, and we got a result of 0.059. And the clinical trial, the real one, had a result of 0.043. So we are quite close within the error bars, and we are quite proud that we were able to reproduce these experimental results purely in silico. And please check our paper for more details. You'll be interested to know that all the software that we did for this project is available in our GitHub, the digital twin, the phantom generation, and the model observer, everything in there. And also, we put all the images of these 3,000 patients. We uploaded to the Cancer Imaging Archive. So if you're looking for easy data for a machine learning training or something like that, this is completely anonymous data because it's not real data. It's virtual data, and you can just download it. And we have actually used this data for machine learning applications. This is a paper we published where we use a faster RCNN network to detect lesions in mammograms. And we train it with clinical mammograms, this DDSM database, and with 500 real images, the network got up to this 0.6 sensitivity. but then we use the simulated data as an augmentation strategy. So we train with 500 real images and 500 synthetic images, and we were able to get a little better sensitivity. And we also use these images for evaluating a denoising software. So we have a very noisy low-dose mammogram here and a high-dose mammogram here. And then we denoise the noisy mammogram with a red CNN denoiser. And we can see with a line profile that, yes, the denoiser denoise the data, but it's not clear if it has any advantage after denoising. That's what we wanted to evaluate. And the way we did was detecting the lesions that we know where they are with the model observer. And we find out that for the mass, really the low dose and high dose and any denoiser didn't have any effect, because the masses occupy many pixels and the people can see through the noise, and the performance didn't change after denoising. However, when you're looking for a tiny microcalcifications, the high dose mammogram had almost perfect detectability, but the noisy mammogram have almost 0.5, so that's no detectability at all. And after denoising, actually the detectability went down. So this is the average image after denoising with red CNN, and the microcalcifications essentially have been blurred and they are not visible. Interestingly, denoising with just a Gaussian convolution did improve a little. So not always the deep learning will be the best approach. All these tools that we are generating and inventing in OCEL, we list them in a catalog of regulatory science tools that you can check out. And also, if you have any tool that can be used for the regulatory process, you can submit them to the FDA using the Medical Device Development Tool Program. And that's a way to, essentially, we review it as if it was a device, but the final product is not a device. It's a tool that companies can use in the regulatory process, and it's a free process, and it's quite rigorous. And you have to select a context of use, and essentially, if the FDA qualifies the tool for a context of use, we are not going to ask questions when this tool is used. So the idea is we don't want to be reviewing evidence for the same thing for every device. If there is a tool that we already trust, then we prefer people use that. But it's voluntary. And the tool doesn't have to be free, of course. It's encouraged to be available, but it doesn't have to be open source or free or anything like that. So in summary, just the main idea is that machine learning applications are regulated as any other software, mostly through a 510K process, but sometimes de novo is common. We need standalone and clinical performance, but will depend on the risk of the software. Consider sending a pre-submission if you have questions. Can be very early. You don't need to develop the device. Can be very early in the development process. And just regulatory science tools available through OCEL and the MDDT program may help you, so you can check them out. Thank you.
Thank you very much, Andrew. So does anyone have want to start with a question for Andrew? If not, I'll start off. So you mentioned that continuous learning for the model training, it's not possible to for this for FDA approved software, or did I hear that wrong? You said continuously updating the software is not possible.
So the rules for continuous learning, they are working on finding the rules, but I think the main point is that the science has not been, it's not there yet. There is the risk, obviously, that any device that changes, once it changes, it can degrade the performance. So the current thinking is that it's still a little early to allow software to be automatically updated. There is some limited cases with de novo where something can be predetermined within the de novo, but it's still early, and in general, it's not possible. And I think it's fair because it's the user of the patient essentially may if the device changes after approval, the patient will be have a device applied on them that has not been reviewed by the FDA, and I'm confident that in the future it could be possible, but at this moment there is no guarantee that the performance will not go down, especially when you reuse. I mean, if you reuse a testing data set many times, that's not right? The performance, it will overfit that testing data set. So you cannot just continuously retest with the training data set and think that 5 years from now the software will be reliable. So we're being a little conservative in this, but the regulations are being developed, so it's still not clear in the future.
Thank you very much. And Farhad, did you want to add something? And Sharina as well?
Sure. Yeah. Thanks, Andrew, for the valuable information. Just to follow up on Eugene's question, first of all, how long this process for FDA approval will take, and if you have something that it goes through updates, let's say version 2.0, how long the second update will take? Is it starting from scratch, or it will be accelerated process?
So the 510K process has by law a limited numbers of days. I will not try to say, but it's about maybe 30 to 90 days. The clock can be stopped at certain moment if there is information needed, but it's quite fast. It doesn't take, especially if there is no issues. It's a small change. It should be quite straightforward. I'm not sure if the people from the companies that talked before may have a more unbiased view, but it's not very time consuming. It's a matter of few months or weeks.
Just one follow-up based on your experience during this process of applying for the FDA approval. What's the main common mistakes that these applications have, and what's your advice for them?
Yeah, so I was happy to hear the previous speakers talking about the importance of the data quality. We don't look at the data. Actually, FDA never looks at the devices. We don't touch the devices. We just read the documentation that is sent to us. So we don't execute the software. We don't look at, the point is we don't look at the training data, but we need like demographics and numbers, like characterizing the training data set. And yes, it's common that we have problems on these. For example, if a segmentation software has been tested only in data from outside the US, that's something that we probably would request that they add maybe up to 50% of the data from the United States, just because the population here, we don't want to be out of distribution, right? That they would say, if it's from a country that doesn't have a different range of obesity and racial makeup, we would want the quality, the data to be from the US. But we would probably, usually we're not so strict with the training. So the training is one thing, but testing is so important for machine learning that essentially we want to know that it generalizes to the population. So we want to see that the Asians and African Americans in the testing data set and the sufficient numbers. And also, if it's for something that uses CT images like segmentation, we want to see a representation of different CT scanners. If it's only one CT scanner, then probably we would ask that the CT scanner is identified in the labeling, saying use it for this CT scanner. But that's not common. Most companies want to use for a range of CT scanners, and that's fine if you have testing in a range of CT scanners.
So who provides this testing data set for you to test?
Not us.
Okay.
Now the sponsor has to provide decide the methods, propose the methods, and that's one of the reasons that I was talking about the pre-submission. For example, if you early on discuss the testing that will be required for your device, then you have maybe a years to get the data. If you go prepare all the device and everything, and then you submit your 510K, and then you find out that we are asking for some data that you don't have, then the 510K will take more time. Then we will stop the time and say, go find that data. So yeah, it's the sponsor who provides the data, and we trust that will be what it's told us it is.
Thank you, Andrew. And Shreem, please go ahead for your question.
So Andrew, I'm a little bit interested in, on students' behalf, what kind of background do people have who work in your department, for example? Who is looking at all of this files who are handed in?
So in the regulatory side, they are all scientists or engineers, a master's degree minimum. I would say most of them have PhDs, biomedical engineering or different kind of degrees. So in my division in particular, we are in the research side. I'm a physicist, and there are several physicists there. We have some mathematicians too that look at the statistics and other like clinical trial design. Statisticians, of course, we have to. A lot of engineers. It depends on the kind of device that we work in. The division of applied mechanics, obviously there's a lot of mechanical engineers. Computer scientists are always appreciated because software is so important. But mostly people looking at the submissions will have master or PhD degree in some science and engineering, and it ideally in the exact kind of device, but that's probably not the case. Probably they need to be trained to the specific device area. So I would say that you can expect like a person that knows about the device. So that we are not afraid of reviewing like, I don't know, anything for machine learning. I'm happy if you send us all the training curves and the loss functions and all the description of the algorithm. We actually request this information usually, like kind of like in a paper or like a diagram of the different blocks and all the skip connections and all these things that you have. And we, since we are scientists that we are publishing in the field, we know the state of the art, right? So we know if somebody tells us about federated learning or like transformer networks, we know what they're talking about. So that's the background.
So you also look at these files in a multidisciplinary group, or it's just one person looking at it, one file at a time?
So there is always a lead reviewer in the regulatory office, not in the Office of Science. And then this lead reviewer assembles a team. It's always a team. The team has a supervisor with more experience, and then depends on the expertise, and they have a system that we send. Like it's kind of an internal consulting system, and if there is a statistics question, there is always a statistician that will look. Almost always there is a medical officer, like a radiologist, whatever discipline is required, that will give a clinical perspective. And then when I come in, it's usually when there is like more technical aspects of medical imaging or deep learning, that it's more like research kind of situation where you need to be at the state of the art. But it's totally multidisciplinary. I mean, we have people from all different specialties. So if there is a specific question, internally we will find someone that has the knowledge.
Thank you.
Thank you very much, Andrew. And that was very insightful for me to listen to as well. I never quite thought about that side, the other side of the work from the regulatory agency side. So thank you so much. And it was very nice meeting you at 8 p.m. as well. So I think that can wrap up our discussion for today. I want to thank all of our speakers again. And we'll see the rest of our participants tomorrow for the workshop. Thank you so much. Have a great rest of the day.
Bye-bye.
Thank you.
Bye.
Bye.